Welcome to our very first episode of this series, Community Conversations with Experts. And our goal here really is to build connections between the research community and the patient community. Open conversations where we can ask really questions that matter to us from the experts who are really doing the hands-on work. And so the intention is to do this on a monthly basis, but we will see how it goes. So my name is Esther Hars. As you know, I have a daughter who has SDS, and I'm the president and CEO of the Shwachman-Diamond Syndrome Alliance and your host for today. Just housekeeping items real quickly. We are recording this session and we may publish it on YouTube and our social media, but you know, your privacy is of really high importance to us, so we will probably remove your video depending on your comfort level for the families. Just let me know if you don't want to be included at all or you want like full-on name and face. That's really a choice you have. Just let me know if you don't want to be visible in the video. We are using the Google Meet platform. The controls are on the bottom, like mute and video, and the chat function is more to the right, a speech bubble looking icon. So we want to encourage questions after Dr. Bezzerri's presentation, and you can both, you know, raise your hand and ask your question on the video, or you can type your question into the speech bubble or the discussion, but I don't know what they call it, the messenger chat, I think they call it chat. And then I can read the question on your behalf. We don't offer medical advice, and we also ask you not to offer medical advice and to adhere to our community standards, which are posted on our website on the bottom. It's basically about having a respectful conversation. Feedback and logistics, about the logistics and the contents of today's session is also welcome, because we're really developing this for you for the maximum benefit of the community as a whole. So if you have ideas and suggestions how to make it even better, let us know. And so without further ado, I would like to introduce Dr. Valentino Bezzerri. He is the head of preclinical research lab at the Cystic Fibrosis Center at the University Hospital of Verona in Italy, and has worked on Ataluren and similar compounds for many, many years, and has been very actively involved in the SDS community for many years. So we are very excited to have you here, and I want to hand over the microphone to you.
Thank you, Esther. Thank you also for the kind invitation to this meeting. I hope this is a really informal meeting, because I prefer informal meetings. I see also some friends as Jacqueline and some colleagues as Giacomo. Giacomo is a new entry of our lab, and is working with us now. Okay. So let me share my screen with you. First of all, I'd like to introduce our team at the Cystic Fibrosis Lab, in particular the Dr. Christian Boni, who is my right arm, actually, and the director of the Cystic Fibrosis Center in Verona and director of the Shwachman-Diamond Center, Dr. Marco Cipolli, and our young collaborators here presented as Elena, Federica, Giacomo is online, and Manuela is another new entry in our lab, who are not present in this photo because it is not updated. But I'd like also to thank all the collaborators that are working with us in this challenging study. And as you can see, a lot of people are working with us, including both Italian and international groups from Cleveland. Professor Seth Corey and his group are helping us in developing Ataluren and also other therapies for SDS. Let me recap briefly the Shwachman-Diamond syndrome. The Shwachman-Diamond syndrome is a genetic disease. That means that it is caused by defects on a gene. In particular, the gene we are studying is SBDS, which is mainly involved in SDS, so in Shwachman-Diamond syndrome. But recently, other genes, including EFL1, SRP54, DNA, GC21, have been reported as associated to the pathogenesis of Shwachman-Diamond syndrome. Shwachman-Diamond syndrome is a bone marrow failure syndrome. That means that bone marrow failed to produce the correct amount of blood cells, in particular the white cells, but also in some cases red cells. So the major issue is at the bone marrow level, but we have to bear in mind that is a pleiotropic disease, so Shwachman-Diamond syndrome involves also pancreas, liver, kidney, bones and teeth, and other organs. It is quite rare as disease, but it is also the most common, or one of the most common, inherited bone marrow failure syndromes. That include also Fanconi anemia, for example, Diamond-Blackfan anemia. So Shwachman-Diamond syndrome is rare, but not as rare as other diseases. We already discussed that the role of SBDS in Shwachman-Diamond syndrome, and now we can discuss why SBDS protein is involved in this syndrome. The SBDS protein has been already widely reported as a ribosomal protein by Professor Alan Warren and his team in Cambridge, UK. And for those who are interested in further details on the mechanism that lead to the generation of mature ribosomes, including the role of SBDS, I suggest to read this very outstanding, I can say, paper by Warren and his group. But at the present time, the take-home message is that ribosomes are the cellular machines that perform protein synthesis. SBDS is involved in ribosome biogenesis, and the efficiency of ribosomes due to loss of SBDS expression or deficiency in SBDS function may lead to altered total protein synthesis, because ribosomes are the cellular machines that perform the protein synthesis within the cells. Looking at the mutations already found in SBDS gene, we can observe that at the present time, more than 20 mutations have been described. But interestingly, more than a half of patients with SDS, either in Italy or outside Italy, have the 182, 183, 184 TA-CT mutation. That is a nonsense mutation. And a nonsense mutation is a mutation that stops the nascent polypeptide chain at the ribosome. That means, sorry, my screen. That means that the formation of the protein is arrested at the mutation point, generating a truncated protein. Fortunately, some compounds, such as Ataluren, reported a read-through efficacy, a read-through effect, so they can override the stop mutation, leading to the generation of the full-length protein at the ribosome site. Ataluren has been already investigated both at preclinical and clinical level in Duchenne muscular dystrophy and cystic fibrosis. In particular, for Duchenne muscular dystrophy, in Europe is approved as therapy. But if we look at the clinical data, so the data obtained from upon the treatment of patients with the drug, we can see here that almost 61% of patients reported a restoration of dystrophin, which is the protein involved in this disease. So Ataluren restored the dystrophin synthesis in almost 61% of muscle biopsies tested. And in this cartoon, it's clear that green protein, so the green fluorescence, means that dystrophin is restored compared to the untreated condition. So these are 2 patients reported with a huge variability in terms of effect. And this variability in terms of effect can be observed also in this graph, in which some patients reported 100% of change in dystrophin expression. Others reported a very low percentage in dystrophin expression. Others, again, reporting no increase at all of dystrophin expression. And this variability is the major limit of the major limitation of this drug. So if we look at the cystic fibrosis studies, or again, clinical studies, we can observe similar results. So 62% of patients reporting an increase of CFTR. In this case, CFTR gene is involved in cystic fibrosis, so CFTR is the target of the therapy, and 62% of CFTR mutated patients reported CFTR synthesis improved in brachial nasal epithelia. Again, looking at the functional effect of these proteins, so the protein is restored as synthesis, but the question was, is it functional? Does it work properly? Ataluren induced nasal chloride transport response, so a functional test, in almost 50% of patients. And if we look at the results, we can observe that patients with the same mutations, in this case the G542X, in one gene, in one copy of the gene, and the F508 delta in the other copy of the gene, so same, we can say, genotype, reported different, very different response to the drug. So this is I'm trying to highlight this limitation because it is very important also for our study. Because we cannot predict that 100% of patients undergoing the therapy will report beneficial effects from Ataluren. But again, if we look at the real-world data in Duchenne muscular dystrophy patients, so we are speaking about thousands of patients tested in more than 10 years of treatment, we can observe 2 major findings: that the drug is not arresting the disease, but is slowing down the loss of ambulation and the loss of lung functions. So it is beneficial, but delaying the age at loss of ambulation and the age at worsening of performance versus the standard of care alone, which is very important, of course. And the second thing is that the second major finding is that in thousands of patients, both adults and children, no particular severe adverse effects were reported. Again, the mechanism of action of this drug has been already widely discussed by Barry Cooperman group in Philadelphia. And they reported that this drug interacted directly with the ribosome, forcing the translation of the full-length proteins, so inducing what we know as read-through. Okay, let's go to our results. So Ataluren in Shwachman-Diamond syndrome. In 2017, we reported a pilot study on 13 SDS bone marrow samples obtained from aspirates gifted by our patients, and we observed that Ataluren restore SDS protein expression in bone marrow stem cells and in white cells in blood, which is important, and improved myelopoiesis. So the leukocytes, the white cells derived from bone marrow, were improved after treatment. And this is an important finding because SDS is renowned as a myeloid syndrome. So the problem, the major issue in SDS in bone marrow is for myelopoiesis. And the drug improved myelopoiesis ex vivo, so outside the human body in cultures. These are the new results recently published in our paper. And as I highlighted here, the study lasts for almost 5 years. We recruited 32 SDS patients carrying non-sex mutations, so the 183, 18040A-CT mutation. And we aspirated their bone marrow, and we cultured bone marrow samples ex vivo, treating cells with Ataluren for 7, 14, or 21 days. And we observed that myelopoiesis, the capacity of producing white cells in bone marrow and therefore then in blood, was improved after Ataluren treatment after 7, 14, or 21 days. On the contrary, the red blood cells were not affected by Ataluren treatment. We discussed before the role of SBDS in ribosome assembly, in ribosome biogenesis. We say that ribosomes are impaired in SDS. So if we can restore SBDS protein synthesis and function, we should obtain a restoration of ribosome assembly. And we tested this hypothesis. This is a graph indicating the formation of the human mature ribosome. This is the higher spike representing the mature human ribosome in healthy controls. And the blue line indicate the untreated cells, the red line indicated the Ataluren treated cells. In this case, no effect from Ataluren, but we can see that the spike of ATS mature ribosome is with a good shape is perfectly sized as a control should be. What if we test some SDS patients? In this case is more evident. The mature form of ribosomes is totally impaired in the absence of Ataluren treatment. Upon Ataluren treatment, red line, we observed a huge restoration of ribosome assembly. With a lesser extent in this patient, and we don't know why, but with lesser extent in this patient, but still improved compared to the untreated condition. So another issue in SDS cells is the accumulation or the upregulation of some harmful proteins, including p53 and others, but we tested at the present time p53, which is a marker for several inherited bone marrow failure syndromes, including Diamond-Blackfan, Fanconi anemia, and Shwachman-Diamond syndrome. We can see that in patients indicated as UPN, because the name, of course, of our patient is totally blinded, so there is a universal patient number. In this case, UPN 58 and UPN 82 reported huge amounts of this protein, which could be harmful for the cells. So we tested a lot of our patients. This is the mean, indicating that p53 is almost upregulated 3 times compared to healthy controls. And what happens when we treat cells with Ataluren? We can observe a reduction of this marker of p53 at levels comparable with the controls. So it seems that has a potential role also in these harmful proteins. And then we sought to investigate the role of Ataluren also in non-hematopoietic cells, including the osteoblasts that are the cells of our bones. So we obtained osteoblasts from bones biopsies of our patients. And in Milan, Isabella Villa, Annalisa Frattini, Simona Bolamperti, Roberto Valli discovered that Ataluren may induce also a re-expression of SDS in bones. And we know that bones is another organ affected by SDS and the loss of SPDS expression. So further details of this study, for those who are interested in further details, I suggest to read the entire work of Cipolla and colleagues, recently published on British Journal of Hematology. I will share with you this paper. Importantly, we are now testing the drug directly in clinical study, so in a compassionate use in 3 patients with SDS followed at the Cystic Fibrosis Center of Verona. Why in compassionate use? Because the company that produces this drug, Ataluren, suggests to initiate the study with a compassionate use, not with a registered clinical trial. A compassionate use is a treatment option that allows the use of an unauthorized medicine, and under strict conditions, products in development can be made available to groups of patients who have disease with no satisfactory authorized therapies, as SDS patients are, and who cannot enter clinical trials. So our 3 patients are adult patients, are undergoing this therapy. 2 of these patients were enrolled almost 5 months ago, and 1 patient was enrolled 2 months ago. The predicted results will be communicated in May 2024. What we can say at the present time is that patients are happy of the therapy and no adverse events were reported in these patients. The problem is that 61, 62 or 50% of patients reported beneficial effects from Ataluren in other clinical studies. So Ataluren might be helpful only for patients with nonsense mutations. This is a first problem, because not all our patients have nonsense mutations. And the efficacy in clinical trial is almost 60%. So we are trying to develop other treats to improve the 60% to 80, 90, or possibly 100% of efficacy. And we are developing analogs with improved efficacy and other therapies for other mutations, including the splicing mutations, which are the most reported mutations in our patients. So if we look at the pipeline we are setting up in our lab, in our center, we can see that a lot of compounds have been already proposed for the treatment of nonsense mutations besides Ataluren, and they can act at different molecular levels. So Ataluren and this other compound may force the read-through by inhibiting this protein. Then there are also other compounds that force the insertion of amino acids at the stop mutation. Other inhibitors of another mechanism that is nonsense mutated decay, but which is not important in this moment. But we are investigating also these drugs that are already available. These compounds are both drugs already approved as Ataluren and Amlexanox, and also compounds in very preclinical conditions, so in very preclinical status. So, for example, the SMG1 inhibitor or the NMD14. These are not drugs, these are just compounds tested in vitro. Then I'd like to show you another study from our group published 1 year ago, in which also this compound, NV848, analog of Ataluren, showed a beneficial role in improving SDS protein synthesis in hematopoietic cells from SDS patients and improving myelopoiesis. The strengths of this compound are that it is completely soluble in water. It already showed an improved efficacy in correcting CFTR protein in cystic fibrosis models in vitro. And again, this compound restores SPTS protein synthesis in SDS cells in vitro, as already said, with outcomes comparable with Ataluren. So, concluding this brief presentation on our studies, we can say that Ataluren showed promising preclinical results that encourage its clinical use in STS. Ataluren efficacy may vary widely regardless of the genotype. So regardless of the type of mutation, so patients with the same mutations, for example, 183, 184, may report different outcomes, unfortunately. Ataluren has been clinically tested on thousands of patients so far, showing a good safety profile, and this is a good point, is a strength of this drug. Other treats are under development in order to treat a larger number of patients. We already have seen the NV848, for example. Other gene editing approaches are under investigation, but in this case, methods need to be improved. So I can stop sharing my screen and I'm ready for all your questions.
Thank you, Valentino. It was very, very interesting. And I particularly like the contrast of where the challenges are and where the opportunities are. Mm-hmm. So to act as kind of an icebreaker, I would like to invite some kids to ask some questions first, and then we'll open up the questions to the rest of you. So let's jump to the questions from the kids.
Why did you decide to become a scientist? Like, what motivated you to do that?
My motivation is to find a cure for somebody. My motivation in research is to do something that may change the life of people. So I'm not interested really in money. I'm not interested in glory. I'm just interested in doing something good for our society. It's like a mission, and this is the first thing I said to all my collaborators or my young researchers in my lab. We have a mission. We have a mission to help other people. I cannot promise you my success, but I can promise that I will do my best to achieve this mission.
Thank you, Valentino.
Do you like work in a laboratory?
Yeah, actually, Giacomo is working in a laboratory. Christian is working in a laboratory. Manuela is working in a laboratory. I'm doing something different. I'm writing projects. I'm writing papers. I'm analyzing data. I'm trying to help young investigators finding the right path to grow as scientists. I'm doing lessons. I'm doing congresses, doing slides every day for presentations. Helping also clinical studies, because in our center we have the opportunity, and this is a real strength for us, to do both preclinical and clinical studies. That means a huge effort from our side, because we have to combine preclinical and clinical issues. But this is also a big opportunity, because it is almost 20 years I'm doing this work. And in the last 10 years, I'm working with Marco Cipolli, the head of the Cystic Fibrosis Center. And his commitment is to do both clinical and preclinical research. And so I'm improving in clinical research. And it is very, very useful for also preclinical, because when you can see the clinical issues, you can go back to the preclinical results and change your mind. When you can know the problems of the patients, the real problems of the patients, they are not just cells, they are patients. So they have more than a molecular problem. And this may change your life. I don't know if you understand the point, but it was totally like a roller coaster for me. Doing this thing is crazy.
Does this medicine require a needle poke?
Does this medicine require an injection, a needle poke?
No, fortunately, this medicine is for oral administration. So it is like,
By mouth,
yes, by mouth,
like Creon?
a drink.
Oh, it's a liquid.
I don't know if the taste is really good, but it's a drink.
Liquid medicine.
What does it taste like?
It tastes of vanilla.
Ooh, like vanilla flavor.
Yes.
That's very cool that you're actually such a concrete medicine that you can describe it so in such concrete terms. That's really
Yes.
cool.
Yeah.
All right, so next, Maria, are you ready for your questions?
Okay. How often would it need to be taken? Would it be every day?
Oh, it was a little quiet. I think you're asking how often one would need to take it, like every day?
Yeah.
Yes. Every day. Every day, 3 times per day. So you have to drink 3 glasses of this drug per day. But also the young patient we are testing is 17 years old. He say that it's not bad. It's not bad at all.
About how long would it be until it's available for people with SDS?
So how long would it take to know whether this will work for patients and for the rest of us, and how soon could it be available for the rest of the patients? I think, is that the question, Maria? Yep.
So the rest of the patients with the nonsense mutations, so ready for Ataluren treatment. This is a good point. First, we have to verify if the drug really works on patients, because it is totally different treating cells in a lab and treating patients in a real life. So once we will be sure that the drug is safe, first of all, because safety is the core of our experimentation, and it works, it really works, do something, improves, I don't know, improves blood cells or improves, reduces the harmful proteins, something like that. We can request a registered clinical trial to extend the use of this drug to other patients all around the world, not all the patients, but a selected number of patients. Then, if also this study will work, we can request to the regulatory agency, so the FDA and the European agency, the approval for the treatment. So, to be honest, it will take some years still.
So several years. So probably like your current compassionate use studies probably will take maybe about a year
Correct?
or so. And then if it looks good, then you would request like a phase 1 or 1-2 trial. So
Super
I
correct.
think the paperwork to get the approval for that will probably, just the paperwork, take many, many months by itself.
Yes.
And
Let-
then
let
the
me-
actual study also, right?
Yes. Let me show a final slide that I was confident that was requested this point. So this is the pipeline, the current pipeline for investigation of starting from a molecule such as NV8048 to the FDA approval, so the commercialization of the drug. It will need, as you say here, more or less 12, 13 years, okay? Starting from the drug discovery to the FDA approval. Using an already approved drug, such as Ataluren or Amlexanox or others, we can skip some of these points, accelerating this process. But this process lasts for, I repeat, 13 years. We cannot reduce the time to 1 or 2 years. We have to spend some years to test. And remember that on 10,000 compounds screened at this at the initial phase, just one arrived at the approval. These are the real numbers.
Mm-hmm.
Ataluren has been already approved, so we can take advantage of repurposing, and this is why we are investigating Ataluren, not just because we are not so brave but because it may accelerate the process of commercialization of this drug. And
So
we hope
basically
it works.
the answer has two parts. Like if everything goes totally smoothly and everything has a good response from the drug, then it will probably take 5 to 10 years. Would you say that's fair?
Less than 10 years.
Less than 10 years. But it will take
5.
at least a few years for approval.
Mm-hmm.
And the caveat is that if there's a good signal that the regulatory agencies consider significant enough. If they don't consider it significant enough, then they will just not approve it.
Yes.
So, and that is unfortunately a real possibility. Even for Duchenne muscular dystrophy, it was approved in Europe, but not in the US.
Looking at the current data, yes.
Yeah.
Mm-hmm. This is a pity because half of our patients, half of general patients in cystic fibrosis and Duchenne reported very beneficial effect. But at the current stages, the FDA approval or the EMA approval will be provided on the basis of a significant effect on the whole population.
Yeah. I think there is a lot of discussions with the FDA and EMA about what it means to have a drug for a rare disease. Because the expectation and the impact should be measured quite differently than in, say, heart medicine that's applied to millions and millions of people. So there is hopefully also going to be an evolution of how the regulatory requirements evolve. But it's tricky. There is really a lot of uncertainty in the regulatory pathways.
Yes. You know, cystic fibrosis is doing the school on this side, because the personalized medicine approach is going on in cystic fibrosis, and we can test if some mutations are responding to Trikafta, the new drug for cystic fibrosis, and request the approval for this specific mutation, adding this mutation in the panel list of mutations already approved. And this is quite new, but yes, these therapies need a different approach, in my opinion, a personalized approach.
And let's see. Emma, would you like to ask a question too?
Okay. If people with SDS took this method, would they go to the hospital less?
So if one day patients can receive these drugs, would that mean that they might have to go to the hospital less often?
I'm not so predictive. I'm not ready
We don't know yet.
to answer this question. Actually, if the drug works, people will be more healthy, and so it means that probably the emergence of complications, and we know the complication of SDS, will be reduced. So probably the story, the history, the natural history of the disease will change as occurred for cystic fibrosis with Trikafta. But in this moment, we cannot predict nothing, because it is, I'm trying always, perhaps Jacqueline knows this point, I'm trying always to be honest. We have to do our best and report the results as they come from the lab or from the clinical words. So
So basically,
unpredictable.
I guess, yeah, we don't know. Basically, it might help with some symptoms and not others, but based on the mechanism of action, it is possible that it will fix many different symptoms. It's possible.
It is possible.
It's not targeting just one particular one, but we just
Yes,
simply don't know at this time.
We are studying Ataluren because we trust in this drug, and we have a good opinion of this drug, but we know that there are limitations, and I reported all the limitations already known for this drug.
All right. And Maria, do you still have more questions, or shall we open it up to the adults?
I have one more.
One more? Okay.
So my sister Nora has a lot of problems with her eyes, like skies, inflammation, and blepharitis. Could the drug help reduce her eye issues?
Was that loud enough? Do you want me to
I can repeat it. So Maria is saying that she has two sisters who have SDS, and one of their problems is all sorts of inflammatory issues around the eyes. And so could this drug potentially help with those kinds more rare symptoms?
So if the drug works properly, yes, it would reduce also inflammation. If drug works.
Yes.
So we know, and we are studying this issue in SDS. Inflammation may be another issue under investigated so far. And Ataluren in vitro, so in our lab and in other labs in Italy and outside, reported also anti-inflammatory properties.
Ramona raised her hand, so let's jump to Ramona's question next. Can you unmute?
There is a question in the chat by Ramona.
Okay.
Can Ataluren be also taken by patients with the DNA JC21 gene? So
Okay.
this is a good question. Ataluren is not targeted to a specific mutation. Ataluren will theoretically correct all the nonsense mutations in all affected genes. So if DNA JC21 gene is affected by a nonsense mutation, yes, Ataluren can be also taken by patients with DNA GC21 mutations. But at the present time, we have never tested the effect of Ataluren in patients with mutation in other genes besides SDS. So it is a longer track, a longer path. Hope to have responded the Ramona question. How are you going to publish the results from the 3 patients? So basically, we will report the data in congresses. We hope at the SDS Congress and perhaps before at the ASH, so the American Society of Hematology Conference. This is our will. If everything goes well and there will be a clinical trial, how are you gonna choose the patients? So far, we have chosen the patients on the basis of the preclinical results. So patients with the good preclinical results were enrolled in our center. And since the company gave the opportunity to test just 3 patients, we selected at random 3 of the most promising patients. I don't know, in a clinical trial, registered clinical trials, especially if it will be international, including US centers, the rules of enrollment. This is a question for Marco Cipolle.
I also wanted to let you know that Dr. Bezzerri has sent me several research articles to forward to you. So if any of you are interested, let me know and I'll forward them to you. As mentioned, I'll, edit this recording for privacy and post it soon. And I guess the last question we can squeeze in. Do you have any idea of what the cost would be like for the treatment?
The drug is currently expensive. A box of drug costs almost $26,000. That is a lot of money.
Is that like per day, per dose, per month?
Month, more or less. Depends on the weight of each patient, because the drug is administered on a weight basis. So we have to know the weight of the patient and then formulate the weight of compound to be administered to the patients. It's not a simple thing to treat with Ataluren. But the patent of this drug by PTC Therapeutics, so the company, is expiring. Yes, next year is the last year of the patent for the formula, so for the production of the powder. And I don't know if this will reduce the costs or not. But currently, the drug is expensive.
And I see 1 or 2 more hands raised. And also, I heard that Emma may have a last question also.
What would happen if they start then stop taking the medicine?
Start and stop. Like, is this something people have to take all the time for work or just for a period of time?
This drug have to be administered always. You cannot stop. You can stop. You can stop, but you will lost the effect of the drug.
And let's, if Valentino, if you have 2 more minutes, maybe we can just take care of the 2 more raised hands. I cannot figure out how to allow people to unmute themselves, even though I changed some settings. So I apologize for that. So currently it looks like we can only take questions in the chat.
Okay.
So unless, I think Jackie was able to unmute herself, but Ramona wasn't. So maybe Ramona, type your question and Jackie can ask her question first. And
Yes.
then we'll have to wind down, unfortunately.
Yes. It is a
Can you
lifelong
hear-
medication,
can you hear
just a short-term medication. It's a lifelong medication, of course.
Right. Can you hear me?
Yes.
Okay. My question was about the iatrogenic effect. Can you explain that?
Tetragenic effect.
This particular medication.
Yes. The drug is not targeted at a single gene. So the drug has a broad effect. It can correct basically all the PTC, all the premature termination codons in all your genome. This may be harmful, of course, this may be a limitation, but at the present time, in all the clinical studies conducted so far, and are quite a lot, treating thousands of patients, also pediatric patients, no particular side effects, excluding rare cases of kidney deficiency, were reported, but rare cases. So I don't know. The main thing is that Ataluren seems to be safe as treatment. The limitation is that the variability in terms of effect. There are patients that feel very, very good with this drug, and others that say no effect at all. So why? This is our future investigation. Why? Because if we can find how it happens, maybe we can improve the drug.
All right, and I see Hannah also just lifted up Amy's question again about the cost, and we covered that. Thank you, Hannah, for noticing that. So I think we are at time, unfortunately, but I'm so glad to see so many questions coming in. And for next time, I'll try to figure out how to allow you to unmute and really speak. But I really wanted to thank you, Valentino, for your time on this Sunday night and your open and honest conversation. If other people have some burning questions, send them to me and I'll forward to Valentino and we can resolve some more questions through emails. And until next time, I hope you'll join us again soon. So thank you. Thank you, everybody.
Thank you all.